Namaste friends, welcome back to Biology Center. We already began the new unit, that is genetics. From that unit we started the chapter Principles of Inheritance and Variation. Principles of Inheritance and Variation chapter. I already uploaded two videos. These videos give you the understanding about the terminology. Unless until you learn the terminology, it is difficult for you to learn the genetic concepts. So it is better to go through the videos and learn the terms. In this video, we talk about very basics. What is genetics? What is inheritance or heredity? Then what are variations? Right. The word genetics was derived from the Greek word. That is Genesis. Genesis means origin of anything, or a beginning, origin of anything or a beginning. Next, the word genetics was given by W. Bateson. W. Bateson was the person who gave the term genetics. Right. Next, definition of genetics. Definition of genetics. Genetics is a branch of biology. Which deals with number one, pattern of inheritance. Number two, gene distribution. Number three, variation. Number four, change in population. Here, these are four areas with which genetics deals. The genetics will explain the pattern of inheritance. Inheritance is nothing but inheritance. The transmission of genetic characters from parents to progeny called inheritance. How that inheritance occurs in organisms, that will be explained by the genetics. The another one is it focuses on the gene distribution. How the genes are segregated and distributed from parents to their progenies. Another one is variation. Genetics also explain us the variation concept. Variation is nothing but differences. In what? The differences in the traits or characters of an individual is called a variation. So finally, the genetics will also talk about whatever changes takes place in a particular population over the period of time. For example, human population. Over the period of time, a lot of changes had been occurring in the human population. How was the first man? Then how is the man at present? Between these two generations, a lot of differences were occurred. So this genetics mainly deals with these four areas. Next, about the meaning of inheritance. So what is inheritance? Right? Inheritance. Inheritance is nothing but transmission of characters from parents to progeny. It is called inheritance of heredity. So during the sexual intercourse, the gametes are transferred from parents to their progenies. So along with the gametes, the certain characters also shift from parents to their offsprings or progeny. Next, variation. So what is variation? As I mentioned now, variation is nothing but differences. Variation is nothing but differences. The differences in characters or traits of individuals of same species. So whatever differences we can see in the traits of organisms belongs to a same species called a variation. For example, all human beings are not in same heights. All human beings do not appear in the same color. The phenotypically and genotypically, the human beings are differentiate from each other. These differences genetically called variations. Variations. For the sake of your knowledge, I tell you the types of variations. Types of variations. Variations are of two types. The first type is called the somatic variation. The second type is a germinal variation. Somatic variation and germinal variation. Actually, somatic variation is non-inheritable. It cannot be inherited from parents to progeny. Germinal variation is the inheritable one. Germinal variation again of two types. The first one is continuous germinal variation. The second type is discontinuous variation. Continuous and discontinuous variation. This continuous variation occurs due to the crossing over, right? Due to the crossing over of sister chromatids, this continuous variation will take place. Then discontinuous variation occurs because of the mutation. Mutation means the sudden change in the gene is called mutation. It is about very brief of the types of variations. Right. Now we are going to know some important prominent scientists contributed their efforts towards the development of genetics field. The first prominent person, so we must remember, was Grigor Johann Mendel. Right. Some prominent scientists contributed their efforts or worked hard for developing the genetics field. The first person was Grigor Johann Mendel. Grigor Johann Mendel. Right. The second person is W. Bateson. W. Bateson. The third person is Morgan. Thomas Hunt Morgan. The fourth person is right, Hugo de Vries. Hugo de Vries. The fifth person is correns. The sixth person is Tschermark. Next, another person is Johansson. Right. First, we talk about Grigor Johann Mendel. He was the father of genetics. For the first time, he laid down the platform for the genetics. He was the first person who did the hybridization experiments. He belongs to Austria nation, right? Place Austria, belongs to Austria. Right. He did hybridizational experiments on Pisum sativa, so which is a garden pea plant. He did experiments from 1856 to 1863. 1856 to 1863, totally 7 years. 7 years. Right. He was the first person, right? So Mendel was the first person who used the mathematics for explaining the biological phenomena. For explaining the biological concepts or phenomena, Mendel used the mathematics. He was the person who maintained the proper statistical records. He did the statistical analysis with accuracy. Right. So of course he gave the postulates, so which help other scientists to carry the further experiments in the genetic field. Next come to Bateson. Bateson was given the term genetics, right? The word genetics was given by the Bateson. He was regarded as a father of modern genetics. Who was the father of modern genetics? W. Bateson. Next, about Morgan. He was regarded as a father of experimental genetics. He did experiments on Drosophila to explain the concepts linkage, sex linkage, crossing over, etc. Next, Hugo de Vries. He belongs to Holland actually. Hugo de Vries, Holland. He did experiments on primrose plant. The another person is Karl Correns. He belongs to Germany. He did experiments on maize. He did several experiments on maize. The other person, Tschermark, he belongs to Austria. He did so many experiments on different flowering plants. Different flowering plants. Then Johann, who called the factors as genes. Actually, Mendel called these genes as factors, right? He called them as factors. The things which are responsible for the expression of characters, called factors by the Mendel. But later on, Johann changed the name and used the term gene. The name given by the Johann. So these are few prominent personalities did so much of work for the development of the field genetics. Here, so after finishing of the experiments by the Mendel, his work was not popularized. So there are some reasons. So let us see the reasons. Right. After 16 years of Mendel's death, three scientists, as I mentioned now, one person is Hugo de Vries, another person is Karl Correns, the another person is Tschermark. The credit of rediscovering of the Mendel work, which was not popularized, goes to these three scientists. As I told you now, so after Mendel's work, that work was not popularized. So because of so many reasons, but after a few years, almost around after 30 years of Mendel's death, these three persons restarted or re-began the work on the same experiments did by the Mendel, then make the Mendel's work more popularized, most popularized. So let us see the reasons for not recognition of the Mendel work. So why Mendel work was not recognized and not popularized? So what are the reasons? So let me write the reasons first. Reasons for not recognition of Mendel work. The first reason is communication. Communication was not proper. In those days, it is very difficult for communicating the information to wide range of population or to the many people. So because of that reason, the work was not published in proper manner. That is the one reason. The second reason is people were unable to understood the factors concept, right? He called the things which are responsible for expression of the characters as factors. So other persons were not in a position to understand the factors. The third one is so other biologists did not accept his work because he was a mathematician. Mendel was not a biologist, actually he was a mathematician. For the results analysis, he applied the mathematics. So that was the first attempt made by the Mendel. So what was the first attempt? Usage of the mathematics for the explanation of the biological phenomenon. So many other biologists did not accept that one. Next, the Mendel did not give any physical evidence for genes and failed in explaining the structure of the genes. So what are genes? How they made up of? These questions, to these questions, so Mendel did not have the answers. These are four main reasons cause the not recognition of Mendel work. Next, come to reasons for success of the Mendel work. How Mendel was succeeded. Reasons for Mendel success. The first point, the first reason is he took a large sample size. When scientists take the large sample size for his experiments, the automatically the issues raised or so demerits raised will almost reduce. So it is one main reason for the Mendel success. The another so reason is data analysis with the accuracy. So whatever data so collected from his experiments that was very neatly analyzed, interpreted and presented. So another reason is he selected contrasting traits. He selected contrasting traits. 7. Contrasting means opposing. So we will talk about contrasting traits selected by the Mendel. So it was also one of the reason for Mendel success. Right. Next. As I mentioned earlier in the beginning of this class, so Mendel was selected garden pea plant, that is Pisum sativum, for his experiments. There are so many plants we know. Why specifically Mendel selected that plant? Are there any reasons? Yes, there are some valuable reasons behind selection of the Pisum sativum plant for his experiments. Let me explain them. Reasons for selection of pea plant. The reasons are very simple. The first reason is Pisum sativum has short life cycle. Pisum sativum plant has short life cycle. It can finish its life cycle in 2 to 3 months because it is a annual plant. Annual plant. Right. The another reason is cultivation. Cultivation of pea plant is easy. The another reason is this plant has many contrasting characters. It is very important to note down. The pea plant has many contrasting characters. Contrasting characters. Then this plant has bisexual flowers, autogamous flowers. Actually, the self-pollination occurs in pea plants naturally, right? Self-pollination is common in pea plant, but so it can be used for cross-pollination also. Mendel actually applied the cross-pollination type for his experiments. Here there is a flexibility. So either go for self-pollination or cross-pollination. Then large number of seeds can be produced. Large number of seeds can be produced in a short period of time. Then seeds do not show dormancy, right? The physical functions are suspended for a period of time is called dormancy. That dormancy is absent in the seeds of a pea plant. So these are the few reasons behind the selection of the pea plant. Right. So finally, I give you one table which represents the contrasting traits selected by Mendel for his experiments. How many contrasting characters are selected by Mendel means 7 contrasting characters. Now I draw the table here. Heading is contrasting characters selected by Mendel for his experiment, for his experiment, right? This is the table I am drawing. So in first column we take serial number 1, 2, 3, 4, 5, 6, then 7. In the second column we take character. In the third column we take traits. First character taken is stem height. Then second character is flower color. The third character is flower position. The fourth character is pod shape. Fifth character is pod color. Sixth character is seed shape. The seventh character is seed color. So these are the 7 characters. So let us see the traits. Stem height. There are 2 traits. One is tall. The second one is dwarf or short. Dwarf is nothing but short plant. The flower color violet. Another one is white color flower. Flower position either axial or terminal position. Pod shape inflated or full. Then constricted. Then pod color green, then yellow, and sheet shape round, then wrinkled. Seed color yellow and green. So characters are 7, traits are 14, right? The list of first line represents the dominant trait, right? All of them are dominant. These list represents the recessive traits, dominant and recessive traits. So while we will have discussion about the monohybrid experiment and dihybrid experiment, so you will get the clarity about dominant and recessive traits or alleles. So these are the contrasting characters or opposing characters selected by Mendel for his experiments. So let me explain these with diagrammatic representation. So it will help you. Tall, this is the tall plant. So this is the dwarf plant, right? This is the dwarf plant. Then color, for example, this is the violet. This is the white color. For example, right? Here, what colors I am using are different. Just then axial. Axial means this is the stem. These are the leaves. So from here, the flowers are right. This is the axial. Then terminal means at the end of the stem. Here, the flowers are bearing at the top. This is the axial. Then inflated pot shape. Inflated means straight, right? Straight. Constricted. Right. This is the constricted shape. Then green color, pod shape is pod color is green color. Then also in a yellow color. Then seed shape, round one. Some seeds are wrinkled shape. Seed color yellow. Some seeds are in a green color. So it is about some basics of the genetics. We will so discuss another aspect in the next video. Thank you very much.